First of all, I want to say hello to Kasia. Thank you very much for accepting our invitation. And also, Giuseppe is here, helping me here today, right, Giuseppe? Thank you very much for coming. Okay, so today, the star of the this afternoon is Kasia Kulej. She's a PhD in, I think the title is Protein Chemistry and Proteomics, isn't it, Kasia? I think so.
It's very close, to be honest. Yeah.
So nowadays she's working at the Children's Hospital of Philadelphia. So it's a great pleasure to have you here. And she's going to talk about some really cool things that she's doing to characterize viral infection, which is actually really, really up to date given the problem that we are all experiencing with this crazy new life that we have. So, yeah.
All right. Thank you so much, Tiago, for this invitation.
Yes.
And of course, thank you all of you who are here today with me to watch it. And so my name is Katarzyna Kulej, and I'm a research associate. I work between two labs, one of Matthew Weitzman from Children's Hospital of Philadelphia, when I primarily perform the biology, and the second lab of Benjamin Garcia from UPenn, when I have a direct access to MS instruments. And today I would like to discuss with you some of the MS approaches which we are using in our labs to study viral infections.
But wait, you you want to start early, but wait a minute, I have something for you. Just 1 minute. Okay? So first, I want to say, as I always say, thank you to just two students that they are in the backstage organizing everything. So, for example, Luis is the one that contacted Kasia, so they're dealing with a lot of things to make this happen. As I already mentioned to you before, yeah, the questions will be in the end of the talk, and you need to write them. So next talk will be from Fabio Gozzo from Unicamp, and later we'll send you guys the title of the talk, but it's going to be next Wednesday, and I think it's going to be the same time, but we will tell you later. Okay, you can
Great.
see using our official media, social media, and YouTube. Kasia, as I mentioned, she already told, so she's a PhD. She got her PhD in Denmark. That's where we got to know her. She's currently in Philadelphia. She'll talk more about that. And just Kasia, we usually show some nice things of how we met each other. So I have some cool pictures that are probably you don't remember.
Oh no, no.
This is an amazing picture, right? It looks like that it's like a picture itself, but this is like a park. You remember that in Germany?
Yes, I remember.
This is when we traveled to for Melanie's wedding. So that's Melanie. So we
Oh
have
my
here
God.
and this is one of the cool parties that we. I don't know if this was in Kasia's place, maybe.
I
And
think yes, indeed. It's my kitchen.
Fabio is hurting Giuseppe here, right, Fabio? So Giuseppe, Fabio, and Kasia and Simone. So here we have like Fight is here. He gave a talk here already. Simone as well. Now it's Kasia, and yeah, and he also gave a talk. Fabio needs to give a talk to us, and yeah, Lee is here. Marcela, yeah, Fabio's wife. Yeah, that's great, right? Do you have these pictures?
Oh my goodness. Okay, guys, now you know me well.
Now you know. Now we know you. Now you can start, Kasia.
So I will use this 5 seconds to actually thank you so much, Louis, who is extremely patient with communicating with me over this last month. So please tell him big thank you for his patience. I'm very grateful. So after the introduction of Tiago, now you really know what I'm doing and who I am, including how looks my kitchen. But okay, so Let's come back to the presentation. So today I would like to show you and explore more of the world of the viruses, and which, as all you know, is extremely large. And especially these days, I don't have to tell you how important it is to study viral infections. And all of the viruses can be divided based on the type of a nucleic acid as a genomic material. And we can distinguish between single-stranded and double-stranded RNA and DNA viruses. And today, particularly, I would like to discuss three viruses, which is adenovirus, herpes virus, and vaccinia. All of these viruses belongs to the group of a double-stranded DNA viruses. So let's quickly compare all of them together. So adenovirus is the smallest one with around 36 kilo base pair of a genome size, following with a herpes virus, which is nearly 4 times bigger, and the biggest one, vaccinia virus, nearly 300 kilo base pair. And adenovirus and herpes virus replicates in the nucleus of the host cell, while the vaccinia replicates in the cytoplasm. All of the viruses leads to contagious and persistent infection of nearly 60-70% of the human population. And especially regarding adenovirus and herpes virus, the infection occurs primarily in the childhood, and infection is a lifelong. Adenovirus and herpes virus can lead to the lytic and the latent stages of infection. And I will show you just here as an example on the HSV. HSV initially infects the epithelial cell as a lytic infection, replicates in the nucleus, and then enters the peripheral neurons when it establishes the latency as a circular DNA. And upon the cell stress, this circular DNA can get reactivated, replicated, and packaged to the viral particle and sent back to the axonal termini, where can actually reinfect back the epithelial cell. And the size of the genome of the viruses can define how many proteins they produce. And this, from other side, defines how dependent or independent are they from the host. In bigger viruses, of course, we would expect bigger independence. And just to compare, the total length of the human genome is over 3 billion base pairs. So you can see how powerful are these little guys. So owing to their very small size, viruses, they have a very restricted life capabilities, and they must infect the host cell in order to replicate, um, uh, uh, propagate its own viral particles. And in order to do so, they developed or evolved to develop strategies to manipulate cell mechanism, not only to control the life cycle, but as well to disable actually the antiviral responses, which are driven through a host immunosystem. So through all of the time course of viral infection, we have this constant battle between the viral and host factors. In Weizmann lab, we are primarily interested in how viruses alter host chromatin regulation. And just to prove you that actually they do so, I'm showing you some of the example of the immunofluorescence stainings of the cells infected with different viruses. And the chromatin is actually labeled by histone proteins, which are the markers of the host chromatin. And just follow with me the white arrows. You can see how the chromatin labeled here in green is changing upon infection. So the viruses, they have this tendency of actually distort or marginalize the host chromatin for its own advantage. They pushing it away to perinuclear areas. And most viruses package their genomes or nucleic acid in the very high molecular densities using a specialized packaging viral proteins to form viral particles. In eukaryotic cells, the DNA is tightly associated with the histones or other proteins to form chromatin. So let's explain first what is a chromatin. The major function of chromatin is to actually package the long DNA molecule into more compact, dense structures. And the chromatin is primarily composed of the DNA and the proteins. The fundamental unit of chromatin is the nucleosome that is composed of around 145 base pair DNA wrapped around the tetramer of the core histone proteins such as H2A, H2B, H4, and H3. And this duplex of this DNA is actually which links two nucleosomes are actually bound to the core histone H1, which serves as a linker histones. And chromatin can be divided on kind of two regions. One which is called as a closed chromatin, more compressed, a silent heterochromatin, and another region which is less condensed, open, so-called active euchromatin. And actually, this high order fold of the heterochromatin is regulated by the post-translation modifications of histone tails, which stick out of the nucleosomes. So let's look closer. What is a histone? So histone tails, or how looks the histone? Histone tails can be very heavily modified by the number of the post-translation modifications from acetylation, methylation, phosphorylation, ubiquitination, crotonylation. I could keep counting. And all of these modifications are regulated by chromatin factors, to which belongs a group of proteins called writers, which add phosphor, add actually PTMs on the histone tails, erasers which remove, and the readers which recognize a specific post-translation modifications. And they recognize by having a very specific domains, like for example, bromo domain of readers recognize the acetylated residues. Within the group of the readers, we can find so-called remodelers, which are the DNA translocases, which they use the energy of ATP to create the force to relocate the nucleosomes. Or for example, they're responsible for the histone turnover on the DNA. And this relocation of the nucleosomes creates the space for the transcriptional machinery to bind to the DNA. So now all of this chromatin factors, which I mentioned here, they can, during active infection, they can go into contact and bind to the nascent viral genomes in order to promote or to inhibit viral DNA replication, gene expression, or other processes required for viral propagation. On the other hand, the recent studies show that infection agents can also largely modify the epigenetic landscape of the host cell by activating or inhibiting histone modifications, by histone mimicry, histone exchange, or by modulating other chromatin binding proteins. So in a Weizmann lab, we ask primarily two questions. So which pathways do viruses hijack to counteract host defense through chromatin? And what we can learn about chromatin dynamics through understanding viral manipulation. But first, to answer these questions, we have to look more deep. So what is on the virus chromatin? What is on the host chromatin during infection? How this translocation is driven through protein-protein interaction or through protein post-translation modifications. So in order to answer these questions, we kind of start building our own toolbox. And of course, we start moving slowly, step by step. So let's start with trying to answer the question, what is on the viral chromatin? In order to look at that, we use a method called IPONT. It's the isolation of proteins on a nascent DNA. So initially, this method was developed by the Cortes lab to analyze the proteins bound to the nascent cellular DNA. And how it works. So during the DNA replications, the analog of thymidine, EDU, gets incorporated into the DNA. And the EDU can be subsequently biotinylated using a click chemistry and the covalent reaction between azide and alkene group. And this biotinylated DNA can be pulled, this DNA together, with, of course, with the protein complexes bound to it, can be pulled down using a streptavidin beads. This method was greatly optimized in my group by my friend Emilio, who did a really great job. So now, primarily regarding IPONT, what I will show is mostly his work, actually. So how looks the full workflow of the IPONT method? So we take as non-infected and infected cells. We label the DNA with an EDU, We cross-link the proteins to the DNA using a formaldehyde. In the next step, we biotin our EDU-labeled DNA. We lyse the cells. We sonicate chromatin on the smaller sizes, and then we use the streptavidin pull-down to capture all of the protein DNA complexes. We are using SDS and boiling to elute proteins from the beads, and of course we send everything for the MS analysis. So here is the time when it's very important to mention that during, especially in the late time points of viral infection, the host DNA replications is nearly completely shut down. So the only replication which occurs supposed to be present on the viral DNA. But to prove that, I will show you here the immunofluorescence staining, where we stain the cells non-infected and infected with different viruses, and we label them for EDU. So we will look where is our incorporated DNA EDU. And in the second panel, you can see the examples of the single-stranded DNA viral binding proteins. So they are labeling the sites of the viral replication centers, so where the viral DNA is present. And you can see that our labeled EDU very nicely overlap with our viral replication centers, which means that a majority of a labeled replicating DNA actually is a viral DNA. The same was for the HSV and for vaccinia. Of course, vaccinia, and I mentioned you in introduction, is replicating in the cytosol, so it's excluded of the nucleosome, which is actually labeled in blue by DAPI here. And to further confirm this observation, we performed the MS analysis of our IP samples, and we wanted to check if all of these proteins are actually indeed the highly enriched in our IP. And indeed, by looking on the fold chain between our samples with biotin and without biotin, which is our negative control for non-specific binding, we can see the very high enrichment, fold enrichment of this protein, including with the rank. So at the last step, we did, of course, the Western blot validation. And indeed, we could, over the time course of infection, after performing an IPOND IP, we actually indeed saw the increase of our marker DNA, viral DNA binding proteins for adenovirus, HSV, and vaccinia. We use pCNA as a control for the host replicating DNA. And in case of the HSV and adenovirus and HSV, we actually see very little traces of it. In vaccinia, you can see actually there is a high enrichment, and indeed we prove that pCNA is incorporated into the replicating vaccinia DNA. So that's why this peak of the signal is present here. Of course, I have to say that there is a certain background coming out from the host, but it's always was always less than 50% after sequencing of the pull-down DNA. So overall, in our approach, we identified nearly 2,000 proteins across our non-infected and infected cells. And we also could track what viral proteins are involved in the replication of the viral DNA. And on my right side, you can see PCA plot, and each black dot represents the replica for certain condition. And actually, this PCA very nicely represents what I also say in introduction, how the size of the viral genome defines how virus is dependent from its host. As I mentioned, adenovirus is the smallest one, so of course we'll use the highest number of the host factors to support its replication, and indeed is the closer to the host. The second is a HSV, and the last, the vaccinia, which is the most independent virus. So this is a very nice picture. So overall, in our pull down, we actually identified proteins which we can divide on three groups. One group of the proteins are the proteins which are equally enriched on the viral and the host chromatin, or there is no significant difference between their enrichment. On the left, in the pink, you can see the number of proteins which actually were significantly more enriched on the viral chromatin compared to host. And these proteins may can have proviral functions in infection. On my right side are the proteins which are the most depleted compared to the host, and they may have antiviral properties. And of course, we have this for all of the three viruses, and then we perform the Go analysis of the pull-down proteins. And indeed, the identified Go terms very nicely reflects the viral cell cycle and the processes which may support the viral replication. I'm not able to describe everything because we have a limited time. So today I will focus primarily on the DNA repair proteins. And to even more narrow the window, I will only explain you what we analyze here for adenovirus. So I want to focus on the proteins which are depleted from the from the adenovirus chromatin. So on my left, I'm showing you the scatter plot, which shows the ratio between the proteins enriched on the adenovirus versus host. And on the y-axis, you have the intensities of the protein. And in the blue, I label the protein which are excluded or underrepresented on the viral chromatin. In red, proteins which are more enriched. And in the middle are the proteins enriched equally or non-significant. And as a control for the proteins which are depleted, we label the proteins which belongs to the mRNA complex. mRNA complex is actually related exactly to the DNA repair, double-stranded DNA repair. And this is our positive control. We know that these proteins are the targets of the E3 ubiquitin ligase complex of adenovirus. And indeed, they are depleted. And here far on the left, we found these three proteins, and we were interested, are there also degradable targets of adenovirus? In order to look at that, we performed the Western blot analysis, when you can see very nicely that over the time course of infection, indeed, the transcription factor is decreasing. We couldn't see this pattern for claspin and ATAD2. It can be that it's maybe antibodies. We didn't go very deep into the validation. So we decided to directly move to the transcription factor 2. And to check if actually this transcription factor can also be degraded similarly to the mRNA complex by the ubiquitin ligase, we made a mutant when we actually deleted the region of the E4 proteins, which are the very important component of the AD5 E3 ubiquitin ligase complex. And indeed, when we remove this protein, so we kind of disrupt this complex, you can very nicely see that we rescue the levels of the TF21 protein. So to ask if it's really the degradation on the protein level, not on the decrease on the mRNA level, we use the proteasomal inhibitor. And you can see that infection in the presence of that inhibitor very nicely rescue the level of the TFT21. So indeed, the degradation is actually on the protein level, so it can be a target of that complex. Here I'm also showing you the IF staining, that you can actually very nicely see that over the time course, the signal of TFT2 during infection in red is decreasing. In the late times, it's creating this very nice foci, which actually are the sign on the proteasomal degradation. And if we will overlap this with the viral replication centers, we can see that TFT2 is actually excluded from that. When we stained with our mutant, the TFT2 is actually overlapping very beautifully with the viral replication centers. So indeed, it's accumulated in our mutant on the viral DNA. And to better specify if which one of these E4 proteins is leading this degradation, we create a set of the mutants, which contains all three proteins, or only two, or only one. And as you can see, the mutant which contains the lack E4 proteins is rescued. But whenever is a lack of a protein, especially E4 or 3, we can also see the increase. But the presence or E4 of 3 proteins very nicely decreases it. And also within a transfection, we prove that overexpression of the single proteins, especially E4 of 3 within a cell, leads to the decrease of TFT21 protein. And indeed, these transcription factors has um, uh, leads as it works as an activator and repressor of a gene expression. And recently it was actually found as well to be responsible for the interferon response upon viral infection. So indeed may have antiviral responses. And of course there is a number of other proteins which we are currently validating, and as well the subset of the group which is retain more on the viral chromatin. And we are looking in all of them, but unfortunately I don't have time to focus on each single today. So I just give you some example to prove that our iPod indeed is working. But what I did as well, I took an iPod and I looked for all of the proteins which are responsible for the regulation of the chromatin structure, and as well proteins which can recognize the histone post-translation modification. And I could fish out really large number of the complexes, histone modifiers, which was pretty amazing. So all of these proteins are actually retained on the viral chromatin. So our next question was, of course, what is happening on the host chromatin and how the histones are regulated. So this is the next step, a next question. So we took another approach, and this time what we decided to do is analyze the entire proteomes in the time course manner. So what we did, we decided to analyze the full proteome of the infected cell, following with its phosphoproteome. In the next step, we performed the chromatin salt fractionation to look primarily on the host chromatin. And of course, we also analyzed the histone PTMs. So we infected the cells, human foreskin fibroblast with HSV, and we collect the samples and different time points. This time point is supposed to reflect the entire HSV cell cycle, from expression of immediately genes to DNA replication, late genes, and then viral packaging. So now I will focus primarily on the proteome. In total, we identified around 5,000 proteins on the host and around 67 HSV proteins. And most of these proteins were very nicely identified through entire 6 time points, and so we can cluster them. And then subsequently, each of these clusters were analyzed for the Go term annotations to kind of check what protein functions are within each of these clusters. And what was amazing is that exactly how nicely all of these clusters could follow the cell cycle again of the virus, from the viral entrance to the cell to the viral replication and viral gene transcription, protein translation, and then a viral release from the cell. So this worked actually pretty well. And beside of the host proteome, we also analyzed the viral proteome. And we can actually see very nicely the trend of the protein expression from immediately early to the late proteins. And actually, our coverage was pretty good. So we identified 67 out of 73 available in the database viral proteins. So that was actually very good. And you can see that over the time of the infection, the viral proteins, which are labeled in red, are slowly increasing in their abundance compared to the host proteins labeled in blue. And you can see that in 15 hours, all of these proteins, they have a higher rank, but they never overcome the abundance of the host proteome, which is obvious, cell would die. So because HSV contains 2 kinases, we ask what's the role of the phosphorylation in infection. And that's the reason why we perform the global phosphoproteome. And as well in that case, we got very nice results. So our depth was pretty large. We identified nearly 12,000 phosphorylation sites on the host proteins and 600 on the viral proteome. And similarly to the on the viral proteome. Similarly, we actually cluster all of these phosphosides. And most of the proteins were identified with a single phosphorylation, but we can also find lots of proteins with multiple phosphorylations. And here I'm just giving example of the host. But what was outstanding here, if you will look on the right, I pointed on the number of phosphorylation for the viral proteins. I just mentioned that the HSV proteome is around 73 proteins, and then nearly one-fourth of it has more than 10 phosphorylation sites, which just show you how probably important these PTMs are in order to drive the viral infections. So maybe that's the reason why actually the virus contains not one but two kinases. And what was also very nice to see that the HSV proteome and phosphoproteome was monotonically increasing over the time course. But what was the difference is that proteome peaked around 12 hours of infection, while the phosphoproteome was delayed and it was peaking very late in infection. So this will also correlates nicely with current actually knowledge that phosphorylation of the viral proteins is primarily important during packaging of the viral DNA, the viral capsid assemble, and also the viral release from the host cell. So this data also very nicely correlate with this what was previously identified. So now we have a proteome and we have a phosphoproteome. So let's go to the next step. Let's look what is on the host chromatin and how it's changing during infection. So how we analyze that? In this case, what we did, we took a cell, we purified the nuclei, and we started washing the nuclei with increasing concentration of salt to wash out the loosely bind proteins and only analyze those which are tightly bound to the chromatin and look how they change in our infections. So that what we analyze is only this last fraction, washed with the highest salt. And what to confirm that in our eyes, primarily what we are looking here is actually the host chromatin. So what we did, we took the two marker proteins, ICP0, which is the HSV ubiquitin ligase, and we know that ICP0 binds to the host chromatin, and ICP8, which is the marker of the viral chromatin, and primarily present on the HSV genome. And indeed, when we look on ICP8, when we wash with the increasing concentration of salt in the last fraction, is nearly gone. So what we assume is exactly that we have primarily here the host chromatin. While ICP0 is slightly present, but it's supposed to be because we know that there is a trace of retain of the ICP0 on the host chromatin. And as well, you can see that histone H3 indeed is increasing in each fraction. And also we don't have much contamination from other cellular proteins. So I cannot say that we don't have a contamination of the viral chromatin here, but in our eyes, primarily what we are looking is the host chromatin. And why as well? Because the viral chromatin, it's more open, more accessible. So it's also easier to wash, of course, the protein complexes compared to the tight chromatin, which can be present in the host. And just show you some of our preliminary results on that. So what I wanted to just highlight here, what we noticed that in infected cells compared to mock, we have a higher retention of the proteins which bind to the acetylated histones, like a bromodomain type of proteins, especially BRD1, which was enriched nearly fourfold change in infected cells compared to mock on the host chromatin. And BRD1 is known to be within the complex with HBO1, CAT7, a histone acetyltransferase. And this acetyltransferase is increasing of the acetylations on the histones. It was for us a bit weird because we would expect that acetylation will increase on the viral chromatin because acetylation leads to more open, accessible for the transcriptional machinery regions. But okay, this is what we received. So we move on with the further analysis. But I would like you to remember these results. So also what we saw in our results and was previously published by a very nice paper in of the Luca lab, that the HDAC protein, so histone deacetylase, is actually retained on the viral chromatin, but nearly excluded completely from the host chromatin during infection. And even the subset of the HDAC2 is actually sent out of the nucleus, which is labeled by DAP in blue, to the cytoplasm. So we can see that we have increase of the histone acetyltransferases from previous slide, and here we have a decrease of the histone deacetylases, which even more promotes the information that indeed we have some increase of acetylome on the host chromatin. Um, so because of these results, we decided to look on the histones. What is happening at that, and indeed, if the all of acetylome is so highly regulated. So analyzing histones during compared to normal proteins is a little bit more challenging. So the histone tails contains load of lysine and arginine residues, and a simple trypsin digestion will create two short peptides to capture them by MS. So what we do, we propionylate to block lysines, so trypsin digest only after arginines, and we get longer peptides. And of course, what we're most interested is to see the PTMs and quantify that. Below, I'm representing just an example of the data which we have from a histone analysis. And for example, for histone H3, so we can quantify a multiple and different type of the PTMs at the same time. And we can define the ratio between them, so how abundant they are relative to non-modified histones. And also what is on our benefit, that actually we can analyze the isobaric forms of the peptides. By isobaric form, I mean it's identical peptides modified with the same number of the PTMs and the same PTM. It's just its position on the different residue. So we can also, with our approaches, distinguish this type of peptides. So let's go to our results. You can see that over the time course, indeed, the histones, PTMs, are getting highly regulated, especially in the late stages of infection. So this is the comparison of the infected cells versus smoke. And what was very nice to see, that our acetylation, especially K9 and K14, indeed showed a very beautiful monotonic increase during infection. And within the group, within the same cluster where these acetylations were found, we also identified phosphorylations on the histone H3 as 10. So we decided to validate them by Western blot, and you can see very nice monotonic increase in all of the cases. We also look on the S28 with similar function to S10. It's very hard to capture by MS due to its low abundance. We verify our observation by IF as well, and you can see that over the time course, indeed, the staining is more bright. And what is interesting that when we look for the ICP8 protein, which stains the viral replication centers, it looks like these acetylations are indeed excluded from the viral replication centers, and they are on the host chromatin primarily in both cases. And just to remind you from the results from my host chromatin bound proteome, indeed, we found this BRD1 and histone acetyltransferases to be more enriched on the host chromatin, and this increases the acetylome on K14, and indeed we see this in histone results, and as well excluding of the HDACs, which can lead to the more global increase of the acetylome on the host chromatin in infected cells. And of course, we also validated the phosphorylations, and we can see that they both increasing, and the same picture, primarily the increase on the host chromatin, and they are kind of looks like it's excluded from the viral. Of course, we cannot really fully say that by IF. We would need to perform the enrichment analysis of this region of chromatin and sequence them, but in our opinion, it's primarily it would be the host. But what was the most striking here is that beside of the increase of the single marks, the peptides, which were the highly actually increasing in abundance, were carrying phosphorylations and repressive marks. So H3K9 trimethyl S10 and H3K27 trimethyl S28 phospho. And this is very interesting because they actually are an example of a process or which is known as a metal phosphoacetyl switch. And I will just explain you what it means. Because I'm more interested of the S28, I will explain this primarily on the H3K27 trimethyl S28 peptide, but below you have a corresponding residues for S10 peptide. So trimethylated K27 is bound by the repressive complex, which keep chromatin transcriptionally silent, not available for like RNA pol 2. And then upon the cell stress, MAP kinases can phosphorylate the neighboring site, S28. This phosphorylation leads to the release of the polycomb. And now this trimethylation is available for histone demethylases and is removed and replaced with by histone acetyltransferases with acetylation. So the phosphorylation creates, we can say, partially open regions of the chromatin, and this acetylation stabilizes the open state and make the chromatin now available for the active transcription. And this process actually, it's already known in the world of the HSV infection. So as I mentioned at the beginning of my presentation, that this circular DNA during a latent infection can get reactivated post-cell stress. And Anna Cliffe showed in her study very beautifully how this process occurs. So the circular DNA is primarily enriched in the repressive heterochromatic marks, which keeps it silent, which is K9 trimethyl, K27 trimethylations. And also stress, a group of MAP kinases is phosphorylating the neighboring residues. This phosphorylation leads to the replacement of the repressive mark with acetylations. And of course, these acetylations leads to a productive reactivation and then a viral gene expression. So, but this is known for the latent infection. So what is doing in epithelial cells during latent infection? And this is still unknown. So we decided to a little bit more follow that topic. A few years ago, there was a very nice study, actually published by a group in Denmark, when they showed that this double mark is primarily dependent from MSK kinase. And they use anisomycin, which induces the cell stress, to prove that the double K27 trimethyl S28 is increasing in the cells, in stressed cells, and as well when they use the H89 inhibitor, which is the inhibitor of the MAP kinase pathway, they could see that it's decreased. And they also show that cell stress indeed activates the phosphorylation of the MSK kinases, and this phosphorylation is primarily the autoactivation sites, so the kinase is active. So we decided to follow up based on this study, and we applied to our model. And indeed, we also induce cell stress using anisomycin, and we also saw the increase in the phosphorylation of MSK, increase in the phosphorylations of the histones. And when we use the inhibitor of MSK, in our case was RMAN64, which is more specific towards primarily the single kinase, we see the decrease in this phosphorylation, including the decrease in the phosphorylation of the S28. And as a control, we also showed the short time course of infection, and in 8 hours we see that S28 is increasing, although we miss here a little bit this band here. And to prove that indeed MSK is activated, I will drive you through another Western blot when we perform the when we use the different concentration of RMM64 to check if actually our phosphorylations will be decreased in infection. And indeed, you can see that in each cases, when we block MSK kinase, our phosphorylations were down, including, of course, phosphorylation as a control of MSK. And what was very interesting is that we don't see a much effect on the early viral proteins, but on the late. So a GC, which is an example, indeed is affected by the blocking of the S20 phosphorylation and blocking of the MSK kinase. To check this, we also validate two other late proteins, TK and VP21, and we saw the same picture. So indeed, the blocking of S28 phosphorylation blocks the late viral gene expression, but not much the early. So maybe there is something expressed out of this host chromatin that supports viral expression. And of course, to follow up that, now we are performing the ChIP-seq analysis to check what is produced from the regions or what is behind these regions of S28 and S10 phosphorylations, and we are trying to follow up how they affect and what's the really mechanism during viral infections. However, how to put all of this information together now? I know it's really a lot and there are different layers of information, and there is no one single experiment which can lead us to explanation of entire signaling pathways or protein functions. So as an example here, just to show you in the one put to one box how we are using these pieces of multiple information together. So as you could saw, you can see I use the proteome to check what is happening to the host proteins. Are they stable in abundance? Are they increasing or they are decreasing in the abundance? Let's say they are stable, like in case of the MSKs, I've checked what is happening with the PTMs. And indeed, some of the proteins they show the increase of the phosphorylation, which may lead to this activation. And in our case was the MSK, which indeed was phosphorylating the histone proteins. And this, again, the phosphorylation of this histone sites leads to increase of the H3 K9 and 14 acetylations. And to prove even this more, we could observe in our chromatin-bound proteome that we have increase of the histone acetyltransferases and bromodomain type of proteins on our chromatin, and we have a decrease of HDACs, which even support more of this increase here. So we need these multiple layers of information to actually fully build the picture of the signaling pathways and how the protein is hijacked by the virus and what's the outcome of it. So to better represent it, I wanted to show you this figure when each of the data set, in our case, leads to different information from protein abundance to protein turnover to protein PTMs to protein localization. And for example, we also analyzed recently using PulseSiLac protein turnover in infected cells and looks really great. Of course, as you saw, I accumulate information about phosphorylation, but also we have currently data sets about ubiquitinations in infections. So what we do, we overlay all of these pieces of information to try to, as best as possible, describe the protein and define what is happening to it through infection. Once we know if the protein changes localization by protein-protein interactions or protein changes in the protein post-translation modifications, we try to mutate, as I show you on the TFT21 protein, certain domains or proteins to check if how they will respond, or you can mutate the phosphorylation site or the another site which is modified by certain PTM and check on the response. So we are analyzing the phenotype. So if upon mutation of this protein, what is affected? Viral entrance, viral replication, viral egress from the host cell. So this exactly defined as the final outcome of a certain signaling pathway or a certain protein. So I know it looks like very time consuming, but unfortunately, that's the best way for characterizing certain pathways through the infection. So just to summarize and not keep you longer, so what we manage is to develop the certain proteomics pipeline to exactly identify the dynamics of the host and viral proteins. We also could define the protein localization on the chromatin virus versus host using an IPON and chromatin fractionation. And using our omics data, we could prove that indeed there are global histone changes and they have a huge impact on the infections. And as well, we show that metal phosphoacetyl switch is a mechanism is very important, not only during latent, but also during the lytic infections, and is driven through a MAP kinase pathway. And currently, to better define the function of this S28 phosphorylation, all of these acetylations, we are performing the ChIP-seq analysis to check what is behind these chromatin domains. And of course, I really would like to thank you to all of my groups, both from Weizmann and Garcia Lab people, for the constant support and help, especially from a biology side, which viral infections are still pretty new to me. Thank you so much, and I'm sorry I keep you pretty long.
No, that was great, Kasha. Thank you very much. Thank you very much. That's like beautiful, beautiful work.
Yeah, we have indeed lots of new data, and I hope that in the future I will show you, especially that I want to really promote here the pulse SILAC. It reveals so much of degradable targets of the virus. And then when we overlay that with the ubiquitin alone, which we achieved, the data are just outstanding. And I really hope that I will share this data soon with you as well.
Yeah. So you're already invited to come again and show us like the new data as
Yeah,
it.
I will be very happy really to show you my other data. And of course, I hope I will show you the follow-up of our S28, which is actually pretty interesting to see that overall is increasing on the host chromatin, has actually huge impact on the viral infection. So we want to know what is behind that. It's just we're working still on that.
So we already have people starting to send some questions. I have a small one, then maybe Giuseppe, and then we can go to the audience. Uh, you did mention, but I didn't catch up about, for example, the viral phosphorylation. Do they also have the kinase, or they hijacked the host kinase to do the phosphorylation?
Actually, it's a good question. The answer is both. So it's very interesting because indeed the viral kinases may directly phosphorylate both host proteins and the viral proteins, and we know already lots of examples. But as well, like for example, it's known that US3 kinase, so one of these two of the HSV, can phosphorylate PKA kinase and activates the downstream entire pathway. So this is also interesting. So actually, the answer is it's actually both.
Okay. And do you know if the protein somehow they have like a more increased affinity towards the kinase to kind of change and balance to like I mean what I want to say is that if somehow the proteins are more phosphorylated, the viral proteins are more phosphorylated by the host kinase compared to the host proteins. Do you know that?
So yeah, so this actually depends on the time course because early in infection, when the viral kinases are very low abundant, primarily things are driven through the host. But
Hmm.
late in infection, when you start accumulating more and more and more viral kinases, they can start taking a lead. Because I think in the end, what is happening, but this is still just my theory, is that everything depends from the time and space. So where are these kinases? What's the abundance? So I still think that it's kind of a competition of the host kinases and viral kinases for the substrates. And now this abundance of these proteins and the localization may actually have a huge impact on the final picture.
Great.
Mhm.
Thank you, Giuseppe.
Uh-huh.
Oh, Kasia, thanks a lot for your talk. It was great.
I'm
Well,
sorry you
I
fast.
already knew most of it, and I can see there are even more news coming up. So I'm really looking forward to see the new stuff you are doing.
Yeah, I have some data that I may share with you indeed. It's just I could keep you guys for like next few hours here. But
No
indeed,
worries.
recently, yeah,
Drago
we
will
had,
invite you next time also.
yeah, we're very happy.
So I was actually, I mean, I think the way you present all of these layers of analysis is a very good lesson to show how we can learn more if we do more.
And
Sometimes,
yeah, I think that, yeah, I agree with you. This is the answer that most of the people they don't like because it's so much of work, so much information to put together. But that's unfortunately it's the way to give a sense of your data.
yeah. And one example of that might be because you did both the total proteome, right? So you analyze at different time points the total proteome, but also you applied this IPON strategy.
Yes.
And when we do the total proteome, well, especially from early time points, the majority of protein do not change. So
Yes,
we get actually most changes during late. But when you apply this iPond, you can see specifically at one specific place, like at this replication, you can see protein that are actually binding to the DNA of the virus. So you can see changes at early time point that maybe, not maybe, for sure, when you do a total proteome, you miss them. So actually, this is another lesson that it's very clear from your presentation that we have to also develop this new strategy. And also, even though the iPond was already shown, as you said, from host protein, like cellular protein, actually you optimize the time of labeling, right? You optimize
yes,
several things to
indeed. I
be
did the
sure
math of
that
that.
you are labeling that.
Mhm.
So my question is, of course, you have applied this to double-strand DNA virus, right? How do you see this technology to other type of virus?
So actually, you have to be indeed
Like,
careful because, yeah, there are also ways to label RNA, and we know
yeah,
that. So instead of like EDU, you will use just different groups. But you have to be sure that, of course, the virus is shutting down the viral host replication. So always, whenever you design, you have to think about what's the background coming out from the host. And so you have to know well how the virus works. So I see that very well for primarily for the viruses which indeed fully shut down the host chromatin. But otherwise, you will get just too much background
from
noise.
your,
Yeah, and as well, it actually worked very well for the, as you could see, for the viruses which replicate in the cytoplasm. Because what we actually observed and was very interesting is that viruses replicating in cytoplasm, they actually steal from nucleus lots of proteins. So they are able to translocate nuclear proteins to the cytoplasm, which is also amazing and actually works very well regarding the iPond. So you have to apply lots of controls, but to know that you have to know your virus. And I know sometimes it's just by trying and failing, yes, and I'm sorry. But regarding your other question, indeed, what is the most difficult during working with viruses is that overall they don't change abundance of the host proteins. They degrade, but some very little number of proteins. The global picture is not changing, so the proteome is stable. What they do, they translocate. They constantly move something around. And that's why we look for the PTMs, which can drive that path. But as well, what is useful, to kind of exactly add another layer for that. That's why is the reason why we apply turnover. Because beside of looking on the turnover of proteins, we also look on the turnover of the PTMs. And sometimes you can see that this PTM indeed is increasing, but the turnover is outstanding. And this is giving you a light, there is something wrong. And it's like pointing you, it's a different layer of information which you would exactly miss from the standard PTM analysis. But you are asking why turnover of this PTM is so high. It's increasing not much, but it's constantly produced and degraded, or add or removed. And this is something one little step more, and can give you a totally different picture on what you are looking for. That's why I kind of really promote as well using the pulsilic for the viral infections works very nice, indeed. And I
another question.
actually,
Oh,
mhm.
sorry, please.
No,
Sorry.
no, no, please continue.
Uh, Tiago, can I ask another question or before moving to? So, Kasia, I mean, I've seen that when you did this phosphoprotein was on the total lysate, right?
Yes.
Do you think it's possible, or have you tried to do a phosphoprotein approach after the iPond?
So I didn't do it. Not I did for histones. So actually, what we did, we used iPond to check the histones bound to the viral chromatin and the histone PTMs. It works. Actually, data were also we did it for HCMV, different virus, and I have to be honest, that looks really great, but you need much more material. So the
Of
difference is that
course.
indeed it works perfectly. It's just we had to increase nearly double the material. But we could do it for histones very nicely. I didn't do the other PTMs. I analyzed histones, and we could see very nice data.
No, it's great. I mean, now also you can combine two techniques and then get even more. Regarding the last question, regarding this interplay between the different PTMs on the histones, well, I really loved the way you show that phosphorylation then blocks, then there is a kinase coming in, then the acetyltransferase. I really like this phosphoacetyl methylation, right? Interplay.
Yes.
When you do that, I mean, of course, you analyze all the histones. I mean, in the laboratory of Garcia lab, this is, I mean, one of the most used techniques. And Simone, of course, was one of them that really pushed this technique forward.
It was me.
I think it's even online watching the talk. But my question was the following. I mean, do you know the kinases all the time that actually can phosphorylate specific residues? I mean, how do you know that of each histone?
So it will depend on the process. So, like for example, Aurora kinase will phosphorylate the histones primarily during the standard the cell cycle. It's a cell cycle related kinase. But MAP kinases phosphorylate these histones only upon cell stress. So indeed that different stress can trigger different kinases. And we also noticed that within MAP kinases, you have R kinases, JNK, and p38. And all of them are triggered by different stressor. So if you stress in different way, different kinase will respond. And there is a difference. It's actually, this is another layer which actually adds difficulties to the analysis. But just in the standard cell, it will be Aurora. Stress cells will be MAP kinases. And now if this will be UV stress, temperature stress, or different, like anisomycin stress, different MAP kinase will respond. And like JNK, if higher response of JNK will lead directly to the cell apoptosis. Why p38 can rescue still cells from apoptosis. So that's why p38 is preferable by a virus, because virus doesn't want cell to die. So actually, what you can see that infection primarily induces the activation of p38, but not JNK. JNK is a late in infection, but then virus wants to get out of the cell, so it allows for the apoptosis.
All right.
So it's another layer of the information and complication.
Yes, thanks a lot.
No problem, Giuseppe.
So we have a question from the audience here, Adriano Pimenta. He actually has two questions, so that's the first. If you or if you have any knowledge of other PTMs in various proteins, and maybe I can already add the next one, because apparently he noticed that the phosphorylation was primarily or mainly in non-structural viral protein.
So what I show the phosphorylation, where of course I show only the proteins which are the most highly enriched. So let me start from question number one. So indeed, there are other PTMs which are found on the viral proteins, and it's a very good question because this part is still not fully explored. I was working with adenovirus proteins when we were actually trying to characterize acetylome. And indeed, the viral proteins are highly acetylated as well. And of course, ubiquitinated, because most of the viruses contains its own A3 ubiquitin ligases. And it's actually surprising that some of the viral proteins also get degraded through time of infection. And the ubiquitin ligases also can label them. Sumomylation is another PTM. And indeed, I never went out of that window. So, but I bet that there is something much more. It's just until now what we're trying to characterize was phosphoproteome, acetylome, ubiquitinolome, and sumomylation. They were primarily the three because of the enzymes which a virus contains. And regarding the second part of the question, it's actually very nice as well, because The structural proteins, I would assume they are the most abundant late in infection, yes? And they are phosphorylated. We also identified plenty of phosphorylation sites. But the proteins which are important from the early time points of infection, primarily I would say non-structural ones, it's those which are last longer. And they can accumulate more modifications because they may perform more functions over this time. But this would be just a theory. While the structural proteins are primarily the late ones, which actually because they're supposed to create the particle, yes, so they accumulate maybe less phosphorylations over this time because they are just nearly when the virus is leaving the cell. But this is just my theory. And I know, for example, that like even the viral kinase of the US3 of the HSV gets phosphorylated over the time course. And this phosphorylation of the viral proteins often defines the localization. Is a nuclear localization or cytosolic localization. And like if a US3 is important in the late time points of infection, it would be phosphorylated, let's say, to enter the nucleus. Where early in infection, it's performing the function primarily outside of nucleus, so it will be there. So it's kind of like I think this PTMs defines exactly where the proteins are. And in longer proteins over the time course of infection, then I think would accumulate maybe more PTMs. But this is just my theory. I have to be honest, I don't know perfect answer for that.
Great. Another question now for Magno. As he said, that's true. 4 projects and 1 big question. Is there any non-recombinase from the host that binds the viral proteins?
It's a good question, and I have to be honest, I cannot answer it. It's out of a kind of like a pool what I'm working with, and I have to say I don't know. I didn't explore that. I'm still in the moving in the virology world, and I'm sorry, I don't know. Mm-hmm.
So yeah, Marcela has a question. This virus kinase could have a big hole on the open chromatin from host.
I damn you! You just hit the most. It's one of the most proper questions actually, because this is what I'm following now. Indeed, what I think is that the viral kinases they want to open host chromatin, and the reason why I still don't know, but it can be that there is something what virus wants to, for example, express to support its infection, or maybe the more silly answer, it wants to open the chromatin for its release from a nucleus, because as we know, the chromatin, especially on the perinuclear surface, it's very tight and compact. So when you relax the chromatin, this allows for the virus to create the channels kind of through the chromatin to leave the cell. And there can be 2 reasons that the opening of chromatin it's maybe beneficial for a virus in the perspective of the viral DNA replication, protein production, or it can be just as silly as creating the tunnels for the virus to leave the cell, for the viral particles. And I'm exploring actually this now. So it's a very, very good question, and we are trying to follow up on that. Mm-hmm.
Great. So Diego, he want to know the software that you use it to interpret your data. So
Oh.
so many, right?
Yeah, it's a actually I had yesterday very nice talk with Martin Krzywiński, who is one of the, in my opinion, best data visualization people. And he also answered there is no single tool. Unfortunately, we use a number of available tools, and I very promote Fight tools. I really enjoy of using his. But as well, of course, Cytoscape. I'm using a lot of Ingenuity Pathway, Metacore softwares, and lots of databases which are available online about the information functions of the PTMs. And we try to pull all of this together. So indeed, there is no one package. I'm just jumping from one tool to another. and I'm just using the best out of it. Sometimes one tool is doing one little thing very good, and I'm just using it for that. And it's a really large number.
Yeah. Another from Magno. How sensitive is the method to the viral proteins in the total proteome?
So actually, we are doing pretty well, and we always can nearly characterize all of the viral proteins.
Yeah.
But there is a one problem, which is like a more a membrane or proteins enriched in glycosylations. Indeed, with these proteins, I think the best is to lyse in like SDS. And when I was performing this, we still didn't could deal much with the detergents for the mass spec. But today we know that S-traps perform so well. So what I would do, I would use S-traps and lyse with SDS instead of urea myself. But usually the sensitivity is very high. The problem is only with a membrane and the proteins enriched with glycosylations. That's why we are losing some of them.
Just a question. Sorry, I'm cutting the queue here, but I mean, have you ever tried to do like an analysis to see the amino acid residues from viral proteins? If the frequency is more or less the same as mammalians, for example, or if they have more sites for PTMs than we do, or
Actually, I think yes, we compare I think tyrosine. We noticed that the HSV contains more tyrosine residues compared to the host.
Okay.
So indeed, there are some amino acids which are preferable by the virus, and in our case, because we analyze phosphorylations, it was a tyrosine, as I remember. We never follow up on that, but this may also state why there can be certain kinases who may be more specific for the viral proteins, because just prefer this residue, yes. And there is some selection of the kinases which targets that amino acid, but I never follow up on that. But this indeed, I think that may be true.
Great. So maybe the last question from Marcelo.
Sure.
Can you read it or?
Yes. Do you think that when the chromatin is open, then maybe the proteins for the replicates are more accessible, but the virus somehow shifts these proteins for its replication? So indeed, it's maybe there is something there, because maybe the HDAC is the good example. Once it because I show you that it's not really on the host chromatin, but it's translocated to the virus. And the lack of this protein, which was stalled by the virus, leads to increase of acetylations, yes? So it can be that, but this is kind of like a after effect. But indeed, maybe the opening of chromatin, of course, make it more accessible for the proteins which prefer accessible regions, while the proteins which binds heterochromatic regions are excluded, yes? So I'm not sure if I can really give an example of that process really here, but it can be indeed.
Yeah, because she says that this links with other questions. If application host shutdown is related with more heterochromatin or block the replication of ill chromatin.
So for sure is we already see that we think it's a less heterochromatin, and I'm trying to go there to show that host chromatin is more open, but I think also there is a less replication of euchromatin. But the reason is so there may be a replication, but the virus is shutting down the production of the host proteins on the translation level. So even if the RNA is produced, it's quickly degraded because it's not really going to translation. So indeed, the replication of the host for sure is affected. But what I think that what I wish to observe, because this is still not explained, I would like to see more euchromatin on the host. But I think it's not entire this chromatin will be actually transcribed.
maybe the last question then. That's Adrian is because he really worked. I mean, when we are studying like PTMs, sometimes we oversee an important PTM, which is protein degradation, right? To
Yeah.
release what's called cryptic peptides, and that's why I think he's asking that. Uh,
So you mentioned that the viral.
so that you mentioned about the viral proteins being degraded in the course of infection, and if you can talk a bit about it, and do you have any experience or data in degradations as PTMs?
So what I regarding viral proteins, there are few examples. I'm sure. Sorry, I cannot really mention that from my head now. But indeed, we saw that some of the proteins they change in abundance slightly in the late time of infections. Although this difference, this degradation of the viral proteins was never dramatic, so still, I would say in non-significant level.
Mhm.
But it can be also that it's hard to also retrieve this information when you have such an abundance of the viral proteins. Yes, but this difference was never really high. There are also other papers from other people and other viruses which they actually try to raise this topic that certain proteins can be degraded. But actually, maybe beside of like degradation of the viral proteins, what is happening that viral proteins are cleaved. So from one protein, and suddenly you have two forms, or a long form and a short form. And a lot of people show that these forms they can have a different functions, or you can have a chimeric proteins of two viral proteins together, or even worse, chimeric proteins of the host and viral protein. And all of this different long short versions they have actually different function. Even us3 kinase is known to have a long and short cleaved version, and there is a few papers show what's the difference in the function of them, that they lack activation of certain substrates or affinity. And regarding the PTM crosstalk with degradation, what I observed that for some proteins, first what we see is the phosphorylation, especially the proteins which are related to heterochromatin. So you can imagine that you have a heterochromatin bound to the chromatin. And of course, the ubiquitinylation is a big PTM. So what is, in my opinion, what is happening that lots of the heterochromatic proteins first gets phosphorylated. Phosphorylation detaches from chromatin. Now they are soluble and they are accessible for the ubiquitin E3 ubiquitin ligases complexes to ubiquitin them and send to proteasomal degradation. So what I think is maybe the steps of the crosstalk of the PTMs, and in my case, I'm trying to work now on some of the chromatic proteins to prove that first they are getting phosphorylated, then they are in the soluble form, and that make them more accessible for degradation. And that I want to see this type of a crosstalk of a, let's say, phospho and ubiquitinylation. But as well, Ileana Cristia lab show very nice crosstalk of acetylone and the phosphorylation regarding lamine. When we have a phosphorylation of lamine following with the acetylations and then a release from the perinuclear membrane. It's also very nice paper.
Great. Yeah. I mean, whole world to be
Yeah,
a right question.
no, what
So,
are really excellent questions indeed. But
I
I'm
mean,
still learning. Like I think all of we, I did not explore all of the fields in the virology. Especially, I'm jumping now more into the RNA and trying to check how viruses are modulating the production of the RNAs and how coding and non-coding RNAs may actually manipulate the viral infection. So this is also huge
yeah, I know.
information. Yeah, so I'm still making little step, and my knowledge is still not full, and I hope one day I will get there.
Great. So thank you very much,
Thank you
Kasia.
so
Was
much,
happy?
guys. It was a pleasure. Mhm.
Yeah. Thanks a lot, Kashi. It was really enlightening.
Oh, we're happy.
This talk.
I'm sorry that it's so long.
It was really nice.
Okay. Thank you so much, guys.
No?
Have a great rest of the day or evening for some.
Yes.
Thank you so much, guys. See ya.
See you. Bye bye. Thank you, everybody. Bye bye. Ciao, ciao.